DNA begins as a double helix and the strands are antiparallel to each other. The nitrogen bases are the ones that are able to form the hydrogen bonds between the opposing DNA strands, which form the twisted ladder effect. The enzyme helicase then enters, breaking down hydrogen bonds, which then opens up DNA. Topoisomerase works ahead of the replication fork to prevent supercoiling. The replication fork is where the DNA splits. There is now an RNA chain which is called a primer, and it works to synthesize the enzyme primase. The primase starts as a contemporary RNA strand and then adds RNA nucleotides along the process. The completed primer is then paired to the template strand. The new DNA strand will start from the 3-carbon end of the RNA primer. DNA polymerase requires RNA primer to initiate DNA synthesis. Remember that DNA is always synthesized in the 5-3 direction. The DNA polymerase synthesizes new strands of DNA continuously of the leading strand and discontinuously on the lagging strand. Remember that the leading strand goes toward the replication fork and the lagging strand goes away from the replication fork. Here, the lagging strand is in fragments, which are called Okazaki fragments. DNA helicase is continuously opening up DNA. DNA ligase then joins the sugar phosphate backbone of all Okazaki fragments into a continuous DNA strand. The DNA ligase is then able to form a phosphate bond between the nucleotides. And finally, the DNA polymerase will go back and proofread for any replication errors.